Small molecule ligands bind to a local structure of their target proteins known as the ligand binding site. These binding events are associated with essential biological functions in living organisms. For example, the binding of the small molecule GTP on the protein Ras-GTPase triggers a signaling event that ultimately controls cell proliferation. When this signaling event is disturbed, it can result in uncontrolled cell growth or cancer. Because of this, scientists are particularly interested in studying the molecular interactions between ligands and their binding sites for drug development. To facilitate this, we have developed a template-based method to design ligand structures for a given target, called STALIS, or Structure Template-Based Ab initio Ligand Design Solution. STALIS uses templates from the PDB libraries of ligands and their fragments to generate a set of virtual ligands that match the shape and chemical features of a given binding site. This video demo will demonstrate the general functionality of the Ligand Designer module in CHARMM-GUI. As the name suggests, Ligand Designer facilitates designing ligands for a given target protein structure using STALIS. The protein structure we'll use is 1-SQT. 1-SQT is a crystal structure for a urokinase protein bound to a 2-naphthamidine-based inhibitor called UI3. Urokinase is implicated in a number of tumor-associated processes, including extracellular matrix degradation and metastasis. Therefore, an effective inhibitor of urokinase can slow tumor growth. We would like to design virtual ligands for this protein. To begin, go to Ligand Designer, type 1-SQT, and click Next. Include both the protein and the ligand, and click Next again. If we want to modify the protein structure, this is where we would do it. To learn how to use the structure manipulation options, please see the video demos for PDB Reader and Manipulator. In this case, we don't need to modify anything, and we also want to keep the disulfide bonds in the protein, so click Next to generate a CHARMM-compatible PDB file. When it finishes, you should view the structure to ensure that it was read correctly. Our structure is now ready to be submitted for GLOSA search in order to design virtual ligands to our choice of a binding site. We offer 4 different ways for users to define a binding site that will be used for ligand designing. Number 1 is to select the crystal ligands and generate a binding site from its surrounding residues. Number 2 is to manually define a binding site by selecting amino acid residues. This can be useful if the query protein structure doesn't contain a bound ligand. Number 3 is to define a binding site by selecting an amino acid residue and specifying a distance from the residue. For example, to create a binding site 10 angstroms around glycine 220, define the radius to be 10 angstroms, then click on residue 220. The fourth option is to predict a binding site using VisGrid software, where you can select which predicted binding site is to be used for ligand designing. In this case, we will use the crystal ligand to define our binding site. This graph shows the estimated running time for GLOSA search that is based on the protein size. Our protein 1-SQT is 244 residues, so it will take about 80 minutes for GLOSA search. When GLOSA search is completed, an email will be sent to the email address that is listed in this box. Don't forget to save your job ID, as you will need this number if you want to retry any steps with Job Retriever. Your job IDs are also available in your user profile. When your job is done, it can be viewed using NGL Viewer. In this result page, we show 2 different tabs for designing virtual ligands from STALIS W and STALIS F respectively. STALIS W designs virtual ligands using ligand templates from a PDB library of whole ligands, whereas STALIS F uses a ligand fragment library. For the urokinase protein 1-SQT, Ligand Designer creates 154 virtual ligands using templates of whole ligands that are ranked using their GA scores, or GLOSA alignment scores. GA scores measure the pocket shape and chemical feature similarity between a virtual ligand binding site template and a target protein's binding pocket. In order to facilitate structural comparisons between our protein and the template protein, we provide their sequence identity to measure the sequence similarity and TM score to measure the global structure similarity. For example, virtual ligand 1 is the same protein as our query protein PDB1-SQT with 100% sequence identity. But if we scroll down, there are some ligands with less than 30% sequence identity that were created by Ligand Designer. For example, virtual ligand 15, chlorophenyltetrazole, that binds to thrombin. The sequence identity between our query protein, urokinase, and thrombin is 29%. Using virtual ligand 15 chlorophenyltetrazole, we can display the target binding site, along with non-covalent interactions, including hydrogen bonds. We can also change the display to show surface representation of the protein to better visualize how the ligand fits in the binding pocket. We can change surface opacity using this slider. Using ligand fragments, Ligand Designer assembles 97 virtual ligands. In the table, we can see the PDB IDs of the structures from which the ligand fragments were taken. For example, virtual ligand 1 comes from 6AG7, 1SQT, and 2VIN, and these are the ligands. In addition to the GA score, we can also add a SLIM score, or shape-based ligand matching, which is a coarse-grained approach of measuring relative binding affinity by calculating shape and chemical feature complementarity between the ligand and the negative image of the binding pocket. A higher score means better fitting of the ligand in the binding pocket. For example, compare virtual ligand 1 with a SLIM score of 3.94 to virtual ligand 97 with a SLIM score of -1.70. In addition to automatically designing virtual ligands, Ligand Designer offers users the ability to manually modify any of the virtual ligands in the list. For example, we can modify virtual ligand 1 by clicking on its modification checkbox, then modify the ligand with a sketchpad powered by ChemAxon. Suppose we want to add another carbon to this terminal methyl group. Our ligand is updated and we can view how the changes are reflected on its 3D structure in the binding site. This can help ensure that your modifications can still fit well in the binding site and prevent steric clashes with the binding site residues. For molecular dynamics and free energy perturbation calculation purposes, we provide ligand parameterization using cgnf. For any selected virtual ligand in the list, you can click Run in the cgnf column to check if a force field for the virtual ligand can be parameterized. If a structure cannot be parameterized due to structural errors or cgnf functional limitations, you can modify the structure with the sketchpad. Note that fragment assembly may yield inappropriate ligand structures, so you should check them carefully. For example, virtual ligand 14 cannot be parameterized. We can alter the sulfide group by removing the hydroxy group, update our structure, and reparameterize it. Once we are satisfied with specific ligands that we want to investigate further, we can download the docked protein-ligand complex structures. In addition, for parameterized virtual ligands, we provide their topology and parameter files that can be used for molecular dynamics system building. In our previous study of protein-ligand simulations, we provided a Python script for automated system building of multiple protein-ligand complexes. In this example, we can use that script to automate system building of our protein-ligand complexes using CHARMM-GUI.